All right, you ready?
I'm ready for it now.
Oh, okay.
55,
Okay. There's 45% to go.
I guess no technology is working today.
And, all right. Well, welcome Dr. Mark Hollier to our fourth STEM talk this semester. And he's going to talk about AIDS and HIV. And he's done a lot of research with the CDC, right?
Yes, I have.
And you can know what he's talking about. So if you'd like to have a popular STEM talk series. Yeah, we have already a few talks set up for next semester, so stay tuned. And yes, you get these links to recordings, and there might also be a link if you want to sign up for the Popular STEM talk community on the JAG list, and you'll find out there. So yeah, without further ado, yeah, why don't you go ahead and get started?
Okay, so I'm going to give a very broad overview about many aspects of HIV and AIDS, starting from its origin through some statistics about how many people are infected, what populations are infected. We're going to look at the virus itself and how it replicates inside cells and how it damages the immune system. And we're going to look at how HIV as a disease progresses in someone who's become infected, to the point that they're defined as having AIDS and what that means to them. Then we're going to have a look briefly at some treatment and some diagnostic tests and a couple of issues with some of those. And then what vaccine research has been done up to about 2-3 years ago. So it's going to be a very broad set of topics that we're going to go over. Hopefully you'll find most of them of interest to you. You characterize as having when you have 200 peak alpha cells per microliter of blood. So it's based on a setback. Nothing that you actually test. What you get infected with is a virus. That's a human immunodeficiency virus, HIV. That's what you would get infected with. That's what you would pass on to someone else if you were to infect someone. You don't catch AIDS. There's a big difference there, which most people seem to confuse with one another. Now, HIV, there are two main types of it, 1 and 2. When you think of the pandemic for HIV, you're pretty good in most cases. HIV-2 is quite localized to one region of Africa, and it's actually, I guess one way of thinking of it is it's slightly milder than HIV-1. It takes, on average, longer for people to progress to the point where their immune system doesn't work anymore. A lot of the signs and symptoms are generally slightly less severe initially, but it doesn't mean they won't catch us eventually, what HIV-1 does. So, you do not catch AIDS. I hear all the time from students around here, it is HIV that is affecting. Now, where it came from, it's been estimated based on genetic analyses that came into the human population around about the 1930s. Now, I'm going to look at the origins of HIV-1 and HIV-2 separately, but they do come from monkeys. Now, there's a lot of viruses and other pathogens that we can catch from other animals, animals other than humans. And they're called a zoonotic disease, that spread from some other animals. Influenza is a good example of that. It's mostly in aquatic birds, and it doesn't seem to have much of an effect on them. But they can pass it on to many other animals. It goes through influenza A, goes through pigs, horses, it can infect humans. So that's another example of zoonotic disease. Now, as I said, HIV comes from monkeys. In monkeys, it's called SIV, simian immunodeficiency virus. And there are different types. And one version from one type of monkey is related to HIV-1. Different version from a different type of monkey is related to HIV-2. So there are some differences. If you want to look at the evidence for when the first person was identified as having HIV, 1959 is the oldest case that has ever been identified. Most people think of the early 1980s when the HIV came to humans, but that isn't true. That was the first group outbreak, which occurred in San Francisco, and I'll be talking about that shortly. Now, one of the problems with how the public became aware of HIV is based on how the media seems to portray it. That led to a big problem with how HIV was actually spread initially. In the early 1980s, it was identified in homosexual men as a disease which was affecting them. It was now identified because there's a couple of diseases which are very rare, unless your immune system isn't functioning. And maybe take a pneumocystis pneumonia. You might get 1 to 2 cases per year across the whole of the US. In the early 1980s, there was an outbreak in homosexual men in San Francisco. So there were several cases which occurred, which was very unusual. So that's when they started looking into it, and they came up with AIDS, acquired immune deficiency syndrome. And they still didn't know what was causing it. They eventually identified the virus HIV as the cause of it. And there's a debate between American scientists about how to name it, because they both claim that they identified it first, but I'm not going to go into that issue today. They was eventually renamed as HIV, so no one really won on that issue. But initially San Francisco. Now, at that time, it was portrayed by the media basically as a white gay man disease. That meant that everyone else figured, well, it doesn't affect me. So they didn't do anything to change their behavior, the pattern of method of how this disease is transmitted. So at that time, the homosexual men did change their lifestyle, and it did reduce the transmission rate. But no one else did. And everyone was still thinking it wasn't going to affect them, because it only affected white homosexual men. So it spread through the population quite badly. The worst affected were the African Americans, especially African American men. There's a number of reasons which have been proposed for that, probably a combination of all of them. At the time, in the mid to late 1980s, the US census data suggests that there was an outbreak of drug abuse in that specific population relative to all the other populations. Sharing of contaminated needles is one way this can be spread. There's also the socioeconomic impact, less education, less access to healthcare, less likely to be identified as infected, and just infecting more people. There's also the general statistics that African Americans generally have more sexual partners than other populations. It's all from the census data. It's all being proposed as being linked. Which one is more relevant? No one can really tell. But they're all sort of combined together. And when we see some future statistics on the slide, you'll see that the African American population is still the most affected compared to any other population in the US. Probably because it had this initial start where there was no control method and it expanded in that population a lot. And then when it changed, it basically reached enough numbers that it kept increasing anyway. So HIV-1. Probably heard in the time there were many reasons proposed as to where HIV-1 came from. The top one aside from God and infection is still being used by certain religious groups today. But there are a bunch of other ones. Now, the polio vaccine. When we make a vaccine is designed to elicit an immune response from a person without actually having the disease. So it's kind of priming it, saying you need to be on the lookout for this. So when someone gets a vaccine, it's not going to cause them disease, but their immune system sees it, and when they do come across it in the real world, they can mount a much stronger, faster response so you don't get disease. The polio vaccine, the live polio vaccine, which is used, sort of discontinued at the moment, but that's another different story. But that one, it has to be grown up in human cells. You have to grow the virus. It's an attenuated version, so it has mutations over time, so it doesn't cause disease anymore. But you're growing it in human cells. Now, if that human cell culture had the virus HIV in it, and you couldn't identify HIV if you didn't know about it, you could potentially transfer it over. That's one problem with live vaccines in general. If they're grown in human cells, you have to really screen that before you use it to make sure there's no other pathogen there. And if you don't know about the pathogen, you have to be screened for it. And even if you do know about it, you have to develop tests to identify it in that sort of time as well. But that was one proposal for it, and as I said, it didn't really turn out to hold any support to it, just like the plague from dogs did. When it comes down to it, HIV-1 comes from chimpanzees. As I said, the SIV, simian immunodeficiency virus. SIV type CTZ for chimpanzees is genetically identical to HIV-1. They have pretty much almost the same sequence. They have the exact same gene in the same order. If you take HIV-2, HIV-2 has a slightly different set of genes. HIV-2 is related to a different SIV virus from City magazine. So HIV-1 and HIV-2 are not identical. They are genetically different viruses from one another. But HIV-1 and SIV-CTZ are the same virus. They have the exact same genes in the same order, pretty much the same frequency. So it's very good genetic data showing that it came from chimpanzees. Now, most zoonotic diseases could be spread through close contact with the animal, could be spread through any type of sharing of bodily fluids that could occur, could be from skinning an animal. There could be exchange of bodily fluids that way. You might get some of the fluid in the air and breathe it in. So there's many ways. One of the more preferred ways that it's thought about is when food gets short, in Africa go and hunt whatever they can catch. Then there's lots of chimpanzees there. So it's thought that if you, just like with lots of diseases, if you don't cook the food properly, some of the pathogens can survive and you could potentially infect yourself that way. So that's a possible proposed method. Now, chimpanzees and humans do have a fairly close association in rural parts of Africa. They do share the same territories, and you do sometimes see chimpanzees walking through the villages as well. So there is some close contact there, which could have led to transmission. How many times it crossed from chimpanzees to humans? No one really knows. Probably more than one. Just have a single transmission to one person to then spread so many ways is fairly low probability if you do the math on it. So it probably had multiple times. Also, if you take a virus from, for example, a chimpanzee and put it into a human, it's not truly optimized to replicate inside human cells, to bind to them and enter them. And they have to adapt. Some viruses cannot adapt, so they cannot cross the species barrier. Other viruses can. And to adapt, comes down to mutations. HIV does mutate very fast due to its genetic zero, which I'll talk about later. Now, HIV-2, this came from city magazine. As I said, HIV-1 and HIV-CPZ are genetically the same. HIV-2 and HIV-SN, the city magazine, is also genetically the same. And it does have a different set of genes to it compared to HIV-1. In addition to having the identical genome, as I said at the start, HIV-2 is more geographically restricted to where it's found. It is really only ever found where the city magazine is. And they have a very small region of Africa, in Western Africa, where they exist. That's pretty much where all cases have occurred. Now, in maybe about 10 years of more international travel, you might get in some other countries, but the majority of it is still found where these city magazines are. Feel free to ask questions as we're going along. So, some statistics. Now, the statistics which I'm going up here are from 2008, so that is about 5 years ago. But in the last 5 years, it has kind of leveled off. So it has been leveling off with new infections, and number of people dying per year and stuff. So if you think about starting in the late 1980s when it was basically unknown, you go from a few cases, and all the way up to 2008, we got to about 33, 34 million people worldwide being infected. So this is just having HIV. The majority of it is in Africa. You're looking at 2/3 of the cases. Now that shouldn't be surprising to you, because that's where the monkeys are, where it was spread from. They also have a much reduced health system, if any at all, and health expenses per year spent on them. They are more likely to go and hunt their food. So there's lots of reasons why Africa is more effective. Now, when we look at the westernized countries, North America, Europe, it is fairly low, and it is remaining fairly stable. Now, the US. When we're looking at the US, these numbers are showing you the number of people per 100,000 who have HIV. Now, just with HIV, and actually pretty much with most sexually transmitted infections, the South is more effective than anywhere else. Now, this again comes back to probably access to healthcare, education level, income level, the proportions of the different ethnicities which are in the South. All of this can make a big difference. And as I said, it's mostly in the South. What always surprises me when I teach the STI chapter in my micro lecture class is that Washington, D.C. always has the highest number for pretty much any STI you can think of, including HIV. I don't know what that says about the politicians, now, if I can get this one to work, this is quite interesting because every time a dot appears, that means there's going to be 50 cases. And you're going to start to see them appearing throughout the map here. Over 2 year separation, so it's increasing. You can see roughly where most cases are occurring. What can you tell me about that?
Major cities.
Major cities. That's where the majority of the cases are. So, you can see Atlanta, got a whole lot over there. Started off over in San Francisco. It's mostly in the metropolitan areas where you find most of the cases. Now, if we look at the ethnicity part, we've got over here the US population from the census data. So you can see, as I said, that it is disproportionate. So over here we're looking at probably about 5% African American in the population overall. But when you look at the percentage of people living with it, African American population is 48%. And that again probably came from how it was portrayed at the start and the lack of control or the occurring at that time in those societies leading to the spread. When you think about the South as well, You have to think about there is probably more of a socioeconomic divide in the South relative to the North and the West. And also that leads to access to healthcare and problems like that. Georgia, it's incredibly hard to find data from the Georgia Public Health Department as how many cases, just pretty much for anything, but managed to find that in the consensus data, 30% African Americans of the state of Georgia, whereas they represent 78% of the cases for having HIV. So it is very disproportionate. This can lead to possibly targeted education on it as a way of reducing it in those populations. But how many people can remember receiving education on HIV and AIDS? Should possibly be done at school level. I'm not sure if it's taught in the school system here, because I never went through a school system here. But who knows? So it's at about 33 million people per year. 2.7 million people were newly infected. So per year, 7,400 per day. And if you look at people dying, about 5,500 people per day. That's worldwide. Now that has leveled off. So it has remained stable for about the last 5 years. That could be because of the increased education on HIV and what is known about how it's transmitted. So it is leveling off. Now, to take the US, we're looking at about 154 people per day being infected and about 63 people per day dying. Those numbers sound high? Oh, relatively high. But if you think of on a state level, you're looking at 50 states. So you'd expect about 3 people per state. In Georgia, 5 people. Again, in the South, higher transmission rate, about 2 people in Georgia per day are dying from HIV and AIDS. Any questions on what we've covered so far?
I guess a question that I have on that is, service and South is considered the Bible Belt, right? I'm sure that some of them have to reflect on religious views where the contraceptives in some cases aren't acceptable alternatives, and which if you're not using any sort of contraceptives in the chance of reducing an STI of any sort,
Yes, it would. That would certainly have an effect. Now, the religious people basically say abstinence and monogamy is all that's needed to control most STIs. But really that is wishful thinking, I guess, is the best way of putting it. It's just not going to happen, really. If you take other STIs, such as gonorrhea has its highest infection rate in 14 to 25-year-olds. These are people who cannot control their hormones, people who don't think about consequences, they have no long-term thoughts about this process, and so it makes it easier to transmit. HIV would also probably fall in that category if you look at it by age. Most STIs get spread in the younger people. And if they go to church, yes, I'm going to be good. So when out at night, who knows what they do? So relative infection, sexual contact is the biggest method of transmission for HIV. Remember, it's HIV that infects you, not AIDS. Now, when you're thinking about that, we started off saying that it was identified in homosexual men in the early 1980s. The major form is heterosexual transmission. And that's based on it being worldwide data. Now, they define the heterosexual transmission as involving high-risk sexual activity. That means multiple partners and protected sex. And most of it, we said 2/3 of it is in Africa. In some African countries, being a homosexual is punishable by death in some of them. So they're not going to identify that as being any way that this disease is transmitted because it doesn't occur. according to the government. In reality, it is a bit, but it's what is, you can only get the data from the government, so you have to go with what they say. The heterosexual transmission is the biggest way of transmitting. The mother-to-child transmission, so this is before, during, or after birth. This is a big issue, and the virus can cross the placenta, so it can go from the mother's blood and cross into the fetus's blood and infect the fetus. It can be the mixing of blood during natural birth and C-section. They can almost be transmitted. And HIV is detectable in breast milk, so it can definitely be transmitted that way. Now, in the last 2-3 years, there's been a lot of work on reducing mother-to-child transmission. If the mother takes the HIV medication and strictly follows the directions for taking it, that mother-to-child transmission can be reduced to almost zero if they take it early enough and they stick to taking it. So that is a very good way of preventing that form of transmission. Contaminated blood, any type of contaminated blood is a possibility. Now, contaminated needles, so you're thinking of drug users, but also people working in the medical field. They give people injections, they can get needle stick injuries if the person jerks their arm and could potentially infect them that way. Blood transfusions is a problem. But if you can't detect HIV in the blood, people have been transfused with it. They do screen the blood, but that depends on what country you go to. If you decide to go abroad shopping for cosmetic surgery because it's much cheaper, you don't know if their blood is being screened as rigorously as over here. You could get any number of diseases that way, and it has been documented that that has occurred. Some countries, the HIV infection rate is extremely high. Anywhere between 20 and even 50% of the population have HIV. One of the countries in Africa is over 50%. Might be Botswana. But when you're looking at that, where are you going to get the blood transfusion? People will die without blood transfusion. So there's much less screening occurring just to meet the demand of blood transfusion. You have to be careful if you have a transfusion abroad, if you work. Same thing with organ transplant. When you think of organ transplant, you have to basically do tissue typing. You have to make sure there's a good match from the donor to the recipient, so that the recipient's immune system doesn't attack and destroy it. Very difficult to find good matches. And if you buy organs on the black market, how do you know if they've been screened or not? But also, in the early 1980s, the tests for screening of HIV, they were non-existent to start with. The first ones developed didn't work particularly well. People still got infected with organ transplant, so they have blood in it. And the last statement here is really to say that if there's more virus in the fluid, you're more likely to become infected. This comes down to, with any infection, one virus particle, one bacterial cell may or may not cause you to get an infection and or the disease. That depends on the specific pathogen. Some of them, yes, one can cause the infection, cause the disease. Others, it requires quite a lot for that to happen. With HIV, a low number will work. No particular organisation wants to give a specific number. All I've ever seen to read is a low number. However, if there's more there, it's more likely that you will become infected and eventually develop AIDS. So, transmission route. Well, first off, you can separate it by the sexes as what the main difference is, and this is just in the US. So, in women, you're looking at the high-risk heterosexual contact. In men, you're looking, more at the homosexual contact as the main method. Now, they might not seem to relate to one another, but there are a lot of males who are bisexual, and they can be exposed homosexually and transmitted heterosexually. If we take globally, heterosexual transmission is by far the biggest form, and as Africa alone represents about 2/3 of the cases, that's the main way they report. If you're looking at any westernized country, well, I've got the data for the US here, you're looking more at homosexual contact, but heterosexual contact is the big proportion really, almost 1/3 in the US. And then you've also got drug use being slightly more of an impact. Now, when you think of transmission doesn't just depend on the number, but there's a lot of physical events that can kill a pathogen. Most STIs require intimate contact. So if they're outside the body for too long, they're dying. HIV is included in that. It will only last about 6 hours, so it's outside the cell. So that's 6 hours from being basically released to being transmitted to the next person and infecting the cell. In reality, it's probably more like 6 hours from one person to another. Outside the body, the temperature goes down. If it dries up, that can damage pathogens and kill them. So there's any number of things which can limit that. Respiratory diseases, they come out of droplets and they travel. But STIs, by definition, they have that intimate contact needed, which really does say that the organism cannot survive outside the host for very long. HIV is actually fairly easy to kill. You can kill it with a little bit of heat for a short amount of time. Just bleach or ethanol is very effective at killing it. Just changing the pH is effective at killing it. It's not a particularly strong virus in the environment. However, if you look at these things, these, if you apply them to persons, also kill the person. So you can't heat them up for 60 degrees centigrade for 30 minutes. So it's easy to stop it when it's on an inanimate object, but it's difficult to get when it's in a person, because what can affect them and affect us as well. So this is a three-dimensional representation of a virus. It's based on what's known structurally about the virus. It has its genetic material here. Now, our genetic material is DNA. Probably all heard of DNA. There's another type of genetic material called RNA. We have RNA as well. In our nucleus, we have a DNA which basically long-term stores the information. When we want to use some of that information, we make an RNA copy, which we send outside the nucleus so it can be read and you can make a protein. That RNA is fairly unstable. It degrades over time. It's just a temporary storage. Now, the genetic material of HIV is RNA. Now, RNA also has a higher mutation rate than DNA. When you copy DNA, the enzyme that copies it reads it and actually proofreads it to make sure it's made a good copy. When you copy from DNA to RNA, or RNA to RNA, there's no proofreading. It copies it and just assumes that it got it correct. And so it has a higher mutation rate. Now that higher mutation rate means that if we make an immune response against HIV and it mutates, it could change what it looks like. Our immune system recognizes specific things it's seen. So if it's now changed its appearance, if it's changed it enough, your immune system will no longer recognize it. It has to start all over again, and the virus is just going to mutate again. So it's a constant battle. Surrounding that, they've got a bunch of proteins. You've got nucleocapsids and capsids. But basically what they are, some of them are actual enzymes that the virus needs when it infects a cell. I'll come to that shortly. And then the capsids of the outer protein part. In general, they're there to protect the virus. Proteins are quite strong in resistance, and it's just holding it all together and protecting it. On the outside, you have a lipid envelope. Some viruses have lipid envelopes, some don't. Lipid envelopes are not particularly resistant to this virus. They can be more easily damaged. So if you take a virus with an envelope, it's much easier to kill it and destroy it than a virus that doesn't need an envelope, just because it's made of lipid. Sticking in the lipid envelope is what they generally term spike proteins. Spike proteins are used to attach to a cell. So it comes along and it uses that spike protein to stick to the cell and to have it gain entry into that cell. That spike protein on HIV, it binds to a molecule called CD4. Now CD4 is on one specific population of your immune cells. And come back to that concept in a little bit. So how the virus comes along and it uses spike proteins to bind to a receptor. And when it bounds to that receptor, it can now trigger itself to go into the cell. Now, what happened right there was the viral membrane fused with the membrane of a cell and ejected the internal part. So that capsid, nuclear capsid, genetic material. Now that's in here. That's inside the cell. Now, HIV is what they term a retrovirus. Normally, the flow of information goes from DNA to RNA to protein. But retroviruses can actually do the RNA to DNA, the reverse process. We cannot do that. We have no equivalent of that. But retroviruses do. So when it gets into the cell, the HIV takes its RNA genome and it copies it into DNA. And then it takes that DNA and it inserts it into your chromosome. So now it's sitting in the chromosome of your cell. Now, you cannot get rid of your chromosome. So it's in there forever. As long as that cell is alive, it's going to be in the chromosome. When the cell divides, it's going to copy its chromosome. Every daughter cell will have it inserted. This is termed a provirus. You used an enzyme called reverse transcriptase to go from RNA to DNA, an enzyme called integrase to integrate it into the chromosome. It can sit here indefinitely. No one really knows what triggers it to become active. So as long as that cell is alive, and every time it copies, it's just sitting there doing nothing. Your immune system cannot see that. Your immune system looks at stuff on the outside of that. And even though you've got the spike proteins outside, they get recycled and removed very quickly. So if it's just sitting in the DNA, there's no way your immune system knows it is there. There's nothing your immune system can do to say that has HIV in its chromosome. So it can never attack and it can never destroy it. Because it doesn't think there's anything wrong. At some point, some of these infected cells will activate. How many? No one can really tell. When? Any number of things could trigger it. Stress could trigger it. Infections from other pathogens could trigger it. The cell dividing could trigger it. There's any number of possibilities that could trigger it. No one really knows. Now, if it does become active, it's going to take that DNA, and if you want to make a protein in your cell, you're going to make an RNA copy of this DNA. The RNA copy is going to come out into the cytoplasm and you're going to make a protein. What happens here is that it wants to make its genome, it just copies that DNA. The DNA remains there. And it can make any number of copies of this. And that RNA is its genome. And it's going to go to the cytoplasm, and you're going to get more and more genomes. And this genome also acts like messenger RNA. When you make a protein, you make messenger RNA, which gets read to make a protein. So when it makes its genome, your enzymes will read that genome and make all the proteins it needs to make its virus particle. Then it's going to undergo an assembly process, and they're going to be released to go and infect the cell. Now, non-envelope viruses, scout the cell, they have to lyse the cell, break it apart to be released. If it's an envelope virus, it can burst out. So this cell can remain producing these viruses for X amount of time. They can produce a lot more over time. Now, as I said, this targets immune cells. HIV can infect macrophages. Now, macrophages are cells that go around. You have a cut. The macrophages are eventually going to get there. They're going to try and eat and destroy anything that shouldn't be in that cut area. any foul debris from your damaged cells is going to try and eat and destroy. So these migrate there. HIV can infect macrophages. But what HIV then ends up going to are your CD4 cells. They're your T helper cells. Your adaptive immune system consists of your B cells and your T cells. Your B cells produce antibodies, which float around all your bodily fluids and will try and bind to anything that shouldn't be in those fluids, as long as it can recognize it. And when it's done that, there's any number of things that conceive antibody and get rid of what's attached to it. Your T cells generally look at your cell, and if you have a virus infecting them, it's going to try and destroy it. T cells can also kill cancer cells if they look different, if they recognize that protein. So you've got one, the T cells looking inside your own cells, and the B cells responding to what's outside your cells. Both the B cells and the T cells, which do those jobs, are controlled by your T helper cells, your CD4 cells. Without the CD4 cells telling them what to do, very few of them are active. Maybe 5% of your B cells will work. And they recognize very specific antigens, very specific targets. And not many organisms have those specific targets. So without your T helper cells, your immune system has no control. It doesn't respond to anything. When this infects the CD4 cell, if it starts producing these, eventually this cell is going to become worn out and die. Or a cytotoxic T cell, one that attacks and destroys the cells, will start seeing some of these coming out and say that's not right. And will attack and destroy it. So ultimately, you end up reducing your CD4 cell number, your T helper cell. That's removing the controllers of the immune system. If you remove the controller, now you can't respond to anything. So any pathogen that comes in could become a life-threatening infection. And that's how HIV ultimately ends up killing you, is it stops you having an immune response, and therefore you end up dying. Now, you still do have your innate immune system. There are some cells called neutrophils, which will see bacteria and try and kill them. There are eosinophils, which will see parasitic worms and try and kill them. There are natural killer cells, which will look at your cells, and if they look a little bit different, they'll try and kill them. But even those need to be enhanced by your T helper cells. And without that enhancement, they don't work to their maximum potential, so ultimately infections end up killing you. So if we take the progression through a person who has just become infected to the day they die, we can split it up into either 4 stages or 3 phases. Now, whether you stage or phase is really dependent on what the country wants to do. Now, if you take stage 1 and 2, they actually represent phase 1. Stage 3 represents phase 2. Stage 4 represents phase 3. So really phase 1 is just these two combined. This first one is a very short period of time. So often you just they turn it into just 3 larger blocks. But I'm going to go through the stages because there is some importance to having stage 1 and looking at what's happening there. So first off, it doesn't last long. And when you become infected, you feel like you get the flu. How many people go to the doctor when they feel like they've got the flu? There's actually a whole bunch of diseases out there which all start out with flu-like symptoms. No one goes. But this is the point where if you started taking the treatment for HIV, all the antiretroviral drugs, you would have the best chance of the infection not establishing itself. Now, you do get an immune response. This graph shows a number of things. This red line represents your T helper cells. So as we said, over time, the virus will infect the T helper cells and it will start killing them off. But over time, as we go through the phases, it gets lower and lower and lower. This black line, which spikes up here and then comes down here, is the number of virus particles in your blood. Now, remember that the virus replicates inside your cell. So once it's inside your cell, it's no longer in your blood. So it's blood on this black line. So initially, you get a big spike of what's in your blood, and at the same time, your CD4 cells really drop down. This is probably because they're becoming infected, they're either being killed by the virus, or you're actually having an immune response and you're killing the infected cells which are producing the virus. Then your number of CD4 cells starts to increase. If you get imbalance in your immune cells, overall your body responds and will try and make more of them. It's a very complex mechanism involved there, but it works. So you recover, you get some more CD4 cells. And the amount of virus in the blood drops down to almost zero. It varies from person to person, but there's very little virus in the blood at that point. So antibodies don't really have much of an effect. This stage is also important because in this short period, that's when the virus population is homogeneous. That means they all look alike. Now, if you're making an immune response, you want to make an immune response to something that all looks alike, so you can attack them all and get rid of them all. If they start to change how they look, then they become heterogeneous. You get mixtures. We said that HIV as an RNA virus, it mutates. One cell, when it mutates in one cell, that's not going to be the same mutation in all other cells. Different mutations will occur in different cells at the same time. So now you get in a mixed population. They all look slightly different from one another. Which ones does your immune system respond to? If it tries to respond to all of them, that's overstretching its resources and its ability. It's not going to work very well. When you enter into stage 2, which we're going to look at in a minute, that's when it becomes heterogeneous. So it's easy to try and eliminate it here, or it would be easy, because they all look the same. Once you get past that, they now all start to look different depending on which cell they're coming from. And so it makes it much harder to respond to them. Another issue in stage 1 is seroconversion. Seroconversion, by definition, means the point at which you start to produce antibodies against the pathogen. So if you become infected and you start producing antibodies 10 days later, in that first 10 days you have no antibodies against that pathogen. 10 days onwards, you do have antibodies against the pathogen. The seroconversion for HIV can be anywhere up to 2 or 3 months before you're producing antibodies. That's very important because when we look initially to see if someone's infected, we look to see if they have antibodies against HIV. If they haven't seroconverted, they're going to come up being told they're HIV negative, even though they have HIV, just because they haven't made antibodies against it. We said 3 or 4 months. Why don't they donate blood in that time? There's no antibodies, no of that blood has got HIV in it. That's the initial sort of test which is done. But seroconversion is a problem because it's 3 or 4 months for HIV typically. Might be less from one person, might be 6, 9 months for another person. It is variable from person to person. So seroconversion poses a problem with donating blood, poses a problem with identifying people as being infected. Moving into stage 2, towards clinically asymptomatic. Asymptomatic means you have no signs or symptoms. You cannot look at someone and say they have that disease. It's very much, they look normal. You don't know that they have any type of disease. Now, in here, you can see that the CD4 cell count, T helper cell, is starting to reduce, starting to decline. So, how long that is depends on the person. Depends on their immune system. Everyone's immune system is slightly genetically different with what they can respond to. So this can be it could be a year, it could be less than a year, it could be 3 or 4 decades. No one really can predict that. So if we watch that CD4 cell count, and we monitor it every few months, we can make a prediction as to how fast they're advancing. Fast progression or slow progression, just by monitoring their CD4 cell count. And that's one of the tests we do when someone's identified as HIV positive. Monitor that to see, give them a prognosis to how long they may have. The HIV itself, very low numbers in the blood. However, you can still infect people. You can infect people in any of these stages. When we go to stage 3, it turns symptomatic. So now you've got signs and symptoms. You can see that if you ask them how they're feeling, they might say, I feel like I got fatigue. They might feel like they're getting a cold. So they're starting to show signs and symptoms. Initially, it's probably general infection ones, which could mean any number of pathogens. But then you get more and more diseases occurring. As this number gets lower and lower for the T helper cells, and the immune system can no longer be controlled as well, you get more what they term opportunistic infection. An opportunistic infection is something that takes advantage. So normally, if you have a good immune system, you won't get opportunistic infection. There are two other ways you could, but for the sake of HIV, that's what you can do. When the immune system's working, your immune system will beat all these organisms, and you'll never know you had that organism in your body. Now, if you're losing control of your immune system because these T helper cells are no longer functioning as well, then these can start to beat your immune system. They might replicate faster than your immune system can respond. So now you start to see these opportunistic infections occurring. Which ones occur? Depends what you've been exposed to, really. You can get any number of viral infections, bacterial infections, fungal, protozoal, anything will start to come up. Because they take opportunity where the immune system is no longer responding. Pneumocystis pneumonia, as I mentioned earlier, is a good example. 1 to 2 cases per year in the whole of the US. If you're immunocompromised, such as HIV, then there's a lot more cases of it in those populations. Because now their immune system, which would normally have seen it and killed it, no longer can do that. So you end up with a life-threatening disease. Pneumonia is just water in the lungs. Can't get enough oxygen in, can't get rid of carbon dioxide. That can also be kill you. So pneumonia can be caused by many things. It's caused by pneumocystis, it's a fungus, pneumonia if you're immunocompromised. At some point, you're going to get down to this line here between stage 3 and stage 4. Now, this is an arbitrary division. We just basically pick when you have 200 cells per milliliter or microliter. When you're looking at that, that is the cutoff point when they say you have AIDS. If you're here versus here, there's really no difference in what you're at. Because there's not much difference between the functioning immune system when you have this many CD4 cells versus that many. So it's just an arbitrary line saying, now you have AIDS. Something we created. So at this point, there's not enough immune cells, T helper cells, for you to make really any immune response or any effective immune response against any pathogen that you come into contact with. So now you start getting a lot more opportunistic infections. Some cancers start to occur more commonly as you approach here and in here. One of them is Kaposi's sarcoma. Very specific, really, for HIV-infected people. But not many people probably develop Kaposi's sarcoma, but your immune cells see it and kill it before it goes more than a few cells. Because your immune system cannot do that, it's not working, then end up with the cancer. So there's any number of things which can go wrong down here. It's actually not HIV itself that kills you. It is the opportunistic infection due to what HIV did to the immune system that ends up killing you. So that's down in here and is 200 cells per milliliter, not micro. Now, with your immune system no longer functioning as well, the amount of virus in the blood starts to increase. Just because there's really no more check to it, it's going to start producing. So I said you can be infected at any stage. In all likelihood, you're probably more likely to infect someone down that end early on because you don't know you're ill. Down here, they've got so many opportunistic infections, they're feeling really bad, that they're less likely to potentially transmit it because if it's a sexual transmission, people are probably not going to want to go near it in that sense. So the virus does increase. Now, to diagnose it, first step I normally do antibodies against HIV. Now, what problem did I say there was with that? Seroconversion. Until you seroconvert, you're not making any antibodies. So from when you get infected to the point of seroconversion, those tests won't say that you have HIV, because of HIV negative. Now, the other problem is specificity. It is not so much a problem today as it was in the 80s. When they first created the test for HIV, they would basically, if you have an antibody against HIV, they would recognize that most of the time. If you had an antibody against another virus, such as HTLV, human T-cell lymphocytic virus, that might have been recognized by that test. They might look very similar. So now you're identifying people who have HIV positive when they're not. Would you want to be told that? So that's the problem with the specificity. Now they have greatly improved on that, but just in general, sometimes antibodies can cross-react. So it's never going to completely go away. It's not that likely to happen, but it's still possible. So now, if you come up as positive with a HIV antibody test, they have to do a second different type of test before they can do it. Now, the ones they can do, they can look for actual antigens. So let's look for antibodies. An antigen is basically a component of the pathogen. So it's just a small piece of the pathogen. So you can look for parts of the HIV virus. If you can detect them, you're sure that that's present, because you've actually detected the virus particle. You can detect its DNA. We can take a blood sample, and in your blood sample you're going to have so many CD4 cells. We can screen those CD4 cells and actually look at their DNA and see if they have the virus inserted in your chromosome. We can't do that in a person, because when we go through that process, those cells are basically dead. But we can actually use it to take a few of them off and see if there's any which are infected or not. So we can look for genetic material. And once we know someone's infected, we're going to monitor what they term the plasma viral load. Now, how much virus is in that load? So it's really low for most of those stages, but they end at increase. We're also going to do a CD4 cell count roughly at the same time. See how fast their numbers are dropping. The faster it's dropping, the less time they have. The longer it takes, the better off they are. So they are for monitoring someone who's infected. Other things can cause CD4 cells to decrease in number. There are other viruses that can do it. So just by monitoring the CD4 cells does not mean that they have HIV. We're just looking at their outcomes once we know they're infected. So plasma viral loads of health, if you're looking for the virus, we can also see how well their bodies responding and controlling. Very important. Antiviral therapies only slow down replication. They do not cure you. There is no cure for HIV. Is there ever going to be a cure for HIV? Hopefully. I personally think it's very unlikely. The reason is, once it inserts into your DNA, it's never coming out. And it could be sitting there for the rest of your life and it could react at any point in the rest of your life. Once it's in that DNA, there's nothing you can do about it. Now, if you think about that, let's one cell got HIV inserted into it, and it just sat there. That cell is sitting somewhere in your body, possibly in one of your lymph nodes. Could be in your leg, could be in your arm. At some point, it might start producing more virus particles, which can go and infect more cells. So if you think that you've reduced it to 0 in the blood of the virus, how do you know about that one cell? With it sitting there. How do you know when it's going to activate? You don't. So when someone becomes infected, I personally think it's unlikely for a cure. Now there have been some reports in the news about people who have been functionally cured. Most of those, basically they had a type of leukemia, cancer of their white blood cells. They had the bone marrow removed. Bone marrow is what produces white blood cells. and they had someone else's bone marrow put in from a donor. Now, if you take out what's producing the white blood cells and you put something else in, you think you've removed all of their white blood cells, which you would get a lot of them. However, there could be some stuff in your lymph node. They could be sitting there for decades before they even leave that lymph node. You haven't removed that one, so they've only been functionally cured. They haven't actually been cured. Who knows when that would activate later, if it would ever activate later. There's no way to go through the whole body and remove every single cell to check it. It's not going to happen.
So, how long is like the person with HIV, like the maximum antibiotics?
So far, there's not one person that's ever recovered from HIV. That's a big problem. And when we look at how we create vaccines, we generally look at how someone who beat the disease, how did they do it? Let's try and mimic that. So that's a big problem with how we create a vaccine. And I will talk about that issue when I get to the vaccine stuff.
So, it's going to have time, like, maybe 2 weeks or
No one knows. It could be sitting there dormant for the rest of the person's life. It might, 2 weeks later, activate, and a lot of them activate, and it progresses really fast. No one knows. Yeah.
So, the way I understood with the whole, when they got the bone marrow transplant, what was going on there was the guy got a, the genetic lottery per se, that very small percent chance. He got a bone marrow transplant that would be resistant to AIDS because it had like what the HIV actually bonds to wasn't on this helper cell, like
wow.
or something to that accord that.
That's what they like to say.
Or
But
to that accord.
I talked about how HIV binds to CD4, a protein receptor. It actually needs co-receptors, right? It binds more than just CD4. Now, some of the co-receptors, one of them is called CCR5. If that has a slight mutation in it, then it makes it very difficult for HIV to get into a cell, but not impossible. If you get a transplant from a bone marrow transplant from someone who has that mutation, it's going to make the cells more resistant. It's not going to completely stop them from being infected. It could certainly slow the process down, but it's not a complete stoppage. Now, there are some reports that there are some people who don't even express that gene. And I think they're investigating right now what that means for the person as a whole if you don't have that gene. That person obviously is alive, but what problems do they face from not having that gene? But theoretically, if there's no huge problem, which cannot be predicted, if you deleted that gene from everyone, it would make it much harder for HIV to get into that. But it does also bind to different co-receptors such as CXCR4. So any cell which has CXCR4 but not CCR5 could be infected. So it's not completely bulletproof in that. It might make it harder, but it might eventually get it.
A question about the serum conversion.
Yeah.
Let's say we have the infection in your chromosome waiting dormant. Would your serum conversion have showed up?
Yes, it would.
Okay.
Well, seroconversion against HIV will eventually happen. It's not going to get into all the cells and just hide and it never happens. It will eventually happen. Whether they get in, some will get in before seroconversion into DNA, others will get in afterwards. So on average, it's about 3 to 4 months.
So it would be detected.
Eventually.
It's not like it's dormant in there and can't be detected ever.
No, it will.
And then
You would eventually detect it, but once it gets in and it goes dormant, over time antibody levels do decline. How fast depends on the specific antibody, really. Not even the pathogen. Some antibodies against the same pathogens disappear much faster out of the blood than others do. Depends what your immune system store on the virus. And it's one of the two, really. So, viruses, they use our own machinery in our cells to replicate themselves. So if we target that, we're going to kill all our cells. So we have to look for unique parts to try and target the use of the treatment. So the ones we can use, we can try and stop it getting into our cells, they're called entry inhibitors. Some of those are currently going through clinical trials. But I don't think there's any which are FDA approved right as of right now. If it's wrong on that, we're not getting close to the end. There's ones which can stop it going from RNA to DNA. So we said reverse transcriptase, an enzyme does that. We have some reverse transcriptase inhibitors. They will stop that from working. So if you don't go from RNA to DNA, you can never insert into the chromosome. We said there's an enzyme called integrase, which takes that DNA and puts it into your chromosome. And it can be any of your chromosomes at any point. It's not saying it's always going to go in this one location. It can go anywhere. And that actually leads to cancer if it puts it in certain genes that control that cycle. So we have integrase inhibitors, which can try and stop that process. We have one called protease inhibitors. Now, when the virus is building itself, it basically ends up building an inactive version. And then it uses one of its own enzymes it makes, called the protease, to activate the virus. It basically has to take that spike protein and cut a little bit of it. Instead of being one big protein, it now becomes two slightly smaller proteins. If that process doesn't happen, it's only one big protein, that virus cannot infect the cell. So if we stop that event, and it's unique to the virus, then we can stop it replicating. And there's research currently going on to try and stop them actually being assembled and or released. These are unique targets we can go after to try and stop them. Now, when we do this, if we only went after protease inhibitors, if we only gave a person protease inhibitors, if it mutates that protein, that drug is now useless. We might have 3 drugs against protease inhibitors, 3 protease inhibitors, 3 different types. One mutation might make it the virus immune to all 3 drugs. If we went after integrase inhibitors, same thing. So what we do is we use what I call a cocktail. You want to take 3 different targets simultaneously. The chance of it mutating its protease, its integrase, and its reverse transcriptase all at the same time is a lot lower than it just mutating one protease. It's possible, but it's much more unlikely. So it's always a cocktail which is used. You go to multiple talents. Now, when I think of medical personnel who end up having needle stick injury and HIV, or they start taking a cocktail of drugs. And if you can take all of these drugs right at the start, you can stop all these events. You're really focusing on these ones here, stopping getting there. But if it does get in there, you want to stop it making more. So you go for all of these, and it's very effective. If you know you've been exposed and you immediately, and we're talking within hours, start taking the medication, it's very effective at stopping you being infected. No. They're not. They're quite expensive. If any drug is made available, the classic example is India. India, you don't get prescriptions. You just go up to a store and tell you any type of antibiotic. They have huge resistance problems because they're taking them irrespective of what disease they have might have no effect. And if you take things which you don't need to take, the microorganisms in your body can start to see it and develop resistance. That resistance can be spread to other organisms, and now that drug doesn't work against anything. Same thing could happen here. So you don't want that situation to happen. If you know you've been exposed, go straight to the emergency room and say you've been exposed to HIV, you need the HIV drug. Another issue, which I don't even know how people came up with this, but there was a big shortage of anti-HIV drugs in South Africa. Turned out people were raiding the warehouses that stored them, stealing them and smoking them to get high. And that led to a shortage of medication for people who were infected, and they would have to get it in Africa. And who knows what outcome that has on the spread for that time period when people can't get their medication. If you stop taking your medication, as soon as you stop taking it, the ones which it was suppressing might start to replicate, and they might mutate against it. So that increases the mutation, and then when you start taking it again, that's resistant. So that's a big problem.
Okay, so that was my question for the healthcare workers that may be infected. What is the time frame? They say usually until the serial conversion when it's infected, or how do you determine how long it would take?
I believe the current standard is a minimum of 6 months.
Okay.
And after that, they're going to keep testing you to see what's happening. But I'm not quite, the standards change country, even I think it changes a bit state by state.
Is that based on the cell conversion though?
Well, not necessarily, no. Because if you're taking these immediately after you're infected, you would possibly seroconvert. You possibly wouldn't. If you stop every virus from replicating and your body broke it down faster than your adaptive immune system to be activated, you would not seroconvert. So what's to say when you stop taking it, you don't go through seroconversion then?
There's some things that, you know, some people like, I don't know, that she takes different guys who have infected on HIV, that you got the virus impact.
I'll come back to that. That's an interesting story we're going to mention later. So there is, it is true. You know, I'd like to why that's not a good thing as well. So, once you start taking these antibiotic therapies, would you want to experience all this every day, all day for the rest of your life? Now, that's the quality of life. If you have to make an informed decision, do I want to even start taking the medication or not? If you start it, and then you decide the side effects are not worth it, you increase the chance of spreading the resistant versions onto other people, and then when they try to take it, it's useless against them. Yeah.
So the way I understand why there's so many typical side effects is because most of these things that you're taking, your cocktails of some sort, are stopping enzymes. And you're not stopping it to that one specific infected cell, but every instance in the body. So anything that uses that is being impeded by the cocktail. And so you're running into a cascade of problems.
Viruses, as I said, they use a lot of our machinery inside them. So, to go from here, it's going to use our DNA polymerases to an RNA polymerases. Once it gets here, it's going to use our ribosomes to make proteins. It's going to use our proteins, our chaperones that fold these and assemble them. So it's using all our machinery. Now, I picked these targets because they're unique. The drug. If we take a AZT, that's one of the original ones which targets the extension of the reverse transcriptase. AZT is preferentially recognized by reverse transcriptase compared to our DNA polymerase. So in an affected cell, the reverse transcriptase is much more likely to use AZT, and therefore the DNA doesn't get copied properly to the virus. So that doesn't mean our DNA polymerase won't occasionally pick it up. And if our DNA polymerase picked it up, it would affect our DNA replication process. So it's a lower chance of affecting ours, but there's a lot more of our cells. Versus, there's a good chance the virus is going to use it and it's going to stop its replication. So a lot of these drugs can cause us problems, which gives us all these side effects. We are targeting unique things in the virus, but because they're using our machinery or enzymes that are very similar to some of our enzymes, it is going to have an effect on us. And that's where you get most of it. Now, as I said, if you stop taking the medication, all the ones that were keeping it back, they've basically now seen it, and they can start to adapt to it. That really is what happens. It's an immune selection process. Even happens with your immune system. Your immune cells for HIV, it kills off all the ones that recognize it. The ones that don't recognize are the ones that survive and take over the population. It's a similar process in both ways. So the problem that mutation of HIV. If your immune system starts to see it, the ones it doesn't see escape. The immune system kills off all the ones it sees, and now you're left with the ones it doesn't see. Now they're around, your immune system has to start all over and start seeing them. It kills off most of them, so it's something it doesn't see, they survive, and it just keeps fighting around. So it's a constant battle. And as I said about the selective advantage, when it's no longer recognized by the immune system or the drug's not working, it takes over the whole population. All of us will be wiped out by the immune system of the drug, but those ones will survive, and now that's all it's left. And they'll have all about themselves to replicate and produce more of them.
It's like we're petri dishes with it. It just keeps going. It stops in one place, it keeps going another place.
It does. It's a constant battle. That's really the same for most infections. It's a constant battle. Problem with this one is that this virus targets our immune system. So the thing that could respond and keep changing eventually doesn't work. So resistant strains, big problem. And I talked about the cocktails as to why we need to use cocktails against HIV to lower that chance of resistance. So I said, once you stop taking it, even if it's just for a few days, that's enough for resistant strains to come through and start taking over. And one person might not be concerned about that, but if they infect someone with that resistant strain, that person will, if they start taking that same drug, will have no effect. Eventually, more resistant strains appear over time, and it takes a long time to make these drugs. It takes the virus a short time to become resistant to it. We don't have the resources or time to keep creating all these over and over and over. There's no resistance. So, vaccine strategy. Before I talk about the actual problems with making a vaccine and the vaccine trials that have occurred, it's important to remember that viruses, they float outside the cell. They're basically dead. They're inert. They don't do anything. They're just floating from one cell to another. Inside the cell, they're replicating. They're actually, could be considered alive. That's still a long, ongoing debate. But they're replicating inside the cell. If we're looking at virus in the blood, you're looking at antibodies binding to it, and then the immune system has a number of ways it can see something that has an antibody to dispose of it. So you're looking at the antibodies against virus in the blood. If it's replicating inside the cell, you're looking at your cytotoxic T cells. They're what infect your cells to see if what's going on inside is normal or not. So you've got two different ways you can try and stimulate your immune system for a vaccine. You can either make it look for something floating in the blood outside of cells, or you can make it aware of, you won't be looking to see if cells look like this. So you can attack and destroy those infected cells. There are two general targets. The antibodies fall under what's termed humoral immunity. The inside cells, infected cells, fall under cell-mediated immunity. If you take a live vaccine, they're very good at stimulating both. If you take a killed vaccine, they're not good at stimulating the cell-mediated one. Yes, cell-mediated one actually has to take inside cells. Also, to create a vaccine takes a long time. You have to go through a number of different phases. So first off, you have the research to find the target, to get enough reproducible data to do that. Someone in another lab is going to have to reproduce it before it's accepted. So it will have to be confirmed by someone else who's related to what you're doing. And then you can go through the phases of that control. That in itself could take 10 years. Once you get here, you're going to give people the vaccine, not to see if it works, but to see if the vaccine is safe. Does putting that vaccine into the person cause a side effect or not? That's what you're looking at. You do it, you get volunteers to do that. It's a low number of people. You're talking 10 for that. Just to see if it's going to cause them to go into suicide, go crazy, a whole bunch of things that is. Once it's shown that it's okay in those tens of people, they're going to put it into 100, maybe low thousands of people to get a better proportion. Tens of people is not representative of the whole population. So now, if you think it's safe, you want to get a better, a more broader spread of ethnicities, ages, and so on, see what effect it has. So here, you're not even looking at if it's effective. There actually, even in low thousands, there's not enough people there to tell if it's effective or not. Then you do that when you go to tens, hundreds of thousands of people, which is a phase 3. People have interpreted this phase 2 data, and it turned out not to be true, just because they didn't have enough people in the study to get the statistics. Now, problems for HIV vaccines are that no one ever beaten HIV. Not one person. Not even Magic Johnson here, that was the time. Now, Magic Johnson's story is that he basically has lots of money. He can afford the best healthcare. He can afford to buy all the latest drugs, which cost lots of money when they've been produced. It is guaranteed that there's going to be no different strains because it's never been used before. But he can afford all of that, and that's probably what's helping him out. Most people cannot do that. Destroys those immune cells. So once your T helper cells are gone, or they're at such a low number, your immune system's not working, your adaptive one's not working almost at all, your innate one is not working at full capacity, so that causes problems. In certain cells, we'll talk about that. If it's in there and it's in our DNA, your body doesn't know that until it starts replicating. So it can be sitting there for X amount of time. You think you've got rid of it all? It's in all about somewhere. That comes back. Not cured. And then HIV has subtypes. Now, we talked about HIV-1 and HIV-2. HIV-1 has different subtypes. A, B, C, D, D, they skip F, turned out to be a combination of A and C, and then there's G, H, N, and O as well. These are where they are so genetically different. So if you take A and B, they're so genetically different from one another that the immune system that recognized A would not recognize B. Would not recognize B. There's enough of a difference that there's no possible chance the immune system would recognize it. So that's a problem. Now, let's also turn clade. Clade A, clade B, clade C. In westernized countries, clade B is the main culprit. If you take Africa, it's A and C. If you take Asia, it's E, and I think it's either A or C. Now, where's all the money to do research? You're looking at, say, big regions. Where are they going to get people who are willing to try it and be paid for it? In Africa and Asia, where it's A, C, and E. How is something which you know won't affect A or C clade going to have you identify if B clade works or not? As a vaccine. So that's a big problem there. But they also, even within clade A or clade B, they mutate. They diverge. They become less and less similar. So that one constantly changes. And there's no good animal models. No. I'm talking about SIV at the start. SIV does not cause the same disease in monkeys that HIV causes in us. In a chimpanzee and a chimpanzee, they can live a full life, have it in their blood, no effect. They might have some minor effects, but it doesn't damage their immune system, it doesn't kill their immune system off like HIV does. But how are we going to test vaccines in an animal model, test drugs, antivirals, if it's not having the same effect on the body? We cannot do that. So that poses a big problem. You can't do human testing. You can't say, I'm going to give you this vaccine and expose you to HIV to see if it's protected. It's not going to happen. So that also creates another problem on its own. You put all these together, chances of getting a good HIV vaccine are looking fairly low. So the vaccine trials which have occurred, and then I come to comment you were making about how people were resistant. So, in this time range, they were basically going against the virus floating in the blood. That's the AIDSvac one. It was against the spike protein. And they basically took two different populations. Now look at their locations. First off, different place with the same vaccine. Probably not going to work anyway. But they're trying two different populations. And the people who received the vaccine versus the people who did not, who received the placebo, there was no difference in infection rate. Now, you cannot expose the people, but you can take people in high-risk groups who are more likely to be exposed to it naturally. Whether they were actually exposed or not, you don't know. Yet there was no difference between the people who were given the vaccine versus the ones who weren't as to how many of them actually were infected with HIV as an outcome. So you're basing it on, we think you might be exposed, so let's try it with you. Are they going to be exposed? You don't know. So it's not even a full trial where you can test it and say, well, we know we exposed you, but you did not become infected. So that comes down to those problems that are really sorted. Now, the next one, which they went for, they said, okay, it doesn't work against the virus, but let's try it, see if we can kill off the infected cells and it's infected. So they took these different locations here, and they tried it. And they actually showed that the people who received the vaccine had a slightly higher chance of being infected than those who did not. That's not a good vaccine. But protection, not making more infected. Now, part of the reason was they put some of the HIV protein into an adenovirus. Now, adenovirus can cause a common cold. So, people that are exposed to it all the time. So you make an immune response against it. Now, if you put some HIV proteins on that, and it's seen the adenovirus previously, it's now going to see the HIV one. Doesn't mean it's going to make neutralizing response. It doesn't mean it's going to be effective. So the response it made somehow enhanced the ability of the virus to get into the cell. The full mechanics of that is not fully understood, but it is somewhat related to they've already had an immune response to adenovirus. Tried a different virus for the adenovirus, one that someone's not normally exposed to, might work better. But it didn't work. It was actually made it worse off for the people who received the vaccine. So what do you think they did? They took both the first two, combined them together and tried them. Two of them failed. Let's stick them together and see what happens. Is that going to work? Does that even make sense? I've never understood why they even attempted that, but they did. Now, they did quite a few people in this, and they took some phase 2 data, analyzed it, and said, it makes a slight difference. It does slightly protect the people who receive the vaccine versus the placebo. Now, statistically speaking, I think it's about 25%, if you get protection of 25% of the population, it's like random chance. They showed 31% in phase 2 data. Now, they never released their data. They just claimed it. And it was right at the time when their research funding was about to expire. So why did they do that? So they got more funding. They did a lot more time on it. And then they showed 24% reduction. Random chance. That's all it was. Now, when they published that data, that led to some people believing there was a vaccine for HIV about to come out. So what do you think that meant?
More activity.
They are careless. And that's certainly something which has been suggested in certain African countries. Where there's a vaccine for HIV, you don't have to worry now. But there's possibly to a spike in infection. But it turned out that it was statistically insignificant. Which you would expect, taking two that don't work, put them together, it probably wouldn't work. Now, the last thing I'm going to talk about is what you were talking about. So, what they noticed was that some prostitutes in Africa, possibly being exposed to HIV, did not get infected. They didn't know why. It could just be the constant exposure, you've got a lot of antibody and it really does stop it if there's a huge amount of antibody there. So what they did was they decided to pay them so that they could study them to see how they were preventing themselves from being infected. Instead of paying them, they stopped being prostitutes. And then they all developed HIV because they were no longer being constantly exposed to it. Yeah. So they were only immune as long as they kept exposing themselves. If they stopped exposing themselves, they lost that protection. We don't even know how that protection works because we can't keep them asking them to expose themselves just to keep looking at them. But no one really knows about that one.
So that means like if one person exposes himself, like
it's not guaranteed. It's not guaranteed that you're going to end up protecting yourself. There's only a small number of prostitutes that occurred in. It certainly wasn't every single prostitute.
It was a kind of like a situation with the serial provider where he said, you know, the antibodies maybe was so high due to the constant exposure that when they stopped being exposed, they thinking it wasn't present and they were able to convert it.
It could be that, yeah. That is one possibility.
Wow.
Yeah. So that's really it. That's a few facts listed here for more information.
that would be like then and you know this like you know anytime you look at this cancer like certain times like cases but like some see them this year.
It varies from person to person. Everyone's immune response is genetically different. Before you were born, your immune system is already programmed as to what it will recognize and what it won't. Everyone has a slightly different genetic program. If they are able to recognize certain protein sequences, which is what that genetic path is based on, if it's a genetic sequence which is HIV, you'll have a cells which can recognize it. Not, you won't. And that's basically recognizing antigen. On one protein, there are a lot of antigens. You take all the proteins on a virus, you're talking about hundreds, thousands of possible antigens. You only respond to a few of them. When you take a bacteria, you're looking probably at tens of thousands or more different antigens. You respond to all of them. What your immune system is genetically programmed to be able to recognize. Now, because of that, you might recognize an epitope that helps you slow down HIV a bit more than someone else. That will give you a better progression as how it goes along, better prognosis. How? We don't have any way of knowing that. So it does vary from person to person. Also, you've got to look at, do you take the drugs, do you not? If you take the drugs, do you strictly follow that prescription and take them all the time or not? At what point did you start taking the drug? Right when you were infected? When you were starting stage 2, towards the end of stage 2? That's unknown as well. All these factors mean you cannot give a predictable time as to how long someone has.
I have a penicillin allergy. It's fairly common. Right?
Yeah.
How does that work with
Penicillin won't work.
Oh, I know that one. I mean, again.
So penicillin targets bacteria, works specifically with mostly gram-positive bacteria. Penicillin stops the cell wall of the bacteria being able to be assembled. So if the bacteria is divided, penicillin could be effective. If the bacteria is just sitting there doing nothing, penicillin won't have any effect because it only stops the building of the cell wall. It doesn't stop it once it's being built. And there's a lot of resistance to penicillin out there. Over time, let's say we use penicillin to treat gonorrhea. It used to be used for treatment. So if we say they used to be used for that, enough resistance built up in the organism that causes gonorrhea that we stop prescribing penicillin to treat them. If our organism is no longer exposed to penicillin, it has no reason to keep that resistance gene. Because to keep copying that gene or making the protein for it uses its energy and resources. So over time, it might lose that resistance, which means we might be able to use penicillin again. Now, that is based on everyone stopping using it against that organism. That's less likely to happen. That's country to country as well. Especially if you can buy any antibiotic you want without prescription. India is notorious for that. And if you do that, people are taking penicillin for anything. They might take it for a common cold. It doesn't work against viruses. But penicillin resistance will develop in the bacteria in that cell.
Biology, but I heard like even bacteria have their own genes, have mutations that they use them for the mechanism, like viruses. Then how do the bacteria break down the genes, the viruses, and so they
We have a similar thing to that, yeah. So, with certain viruses, when they infect our cells can produce some enzymes which can break down their RNA, if it's an RNA genome virus. And it can also produce it basically stops our cells from making proteins. Now, our cells can survive so long before it has to restart making proteins. So it might be long enough to stop from that, the virus cannot replicate, and the RNA genome has now been broken down during that time. It doesn't work against all viruses. The same thing doesn't work against all bacterial viruses, which are called phage, when that happens in them. So we have defense mechanisms. But viruses, they replicate much faster than we do. How long does it take for someone to have offspring? Let's say, theoretically, puberty. So let's say, that age is going up, let's say 10 to 14 years old. How long does it take a bacteria to divide? If you look at E. coli, optimally you can do it in 20 minutes. They can divide, and every time you divide, you're going through basically evolution. More division, the shorter the evolutionary time is needed to get changes. So when you're looking in respect to viruses, it's a very similar thing. They can replicate so much faster and incorporate errors, mutations at a much faster rate than we can. So they can basically evolve faster than we can.
kind of virus do you
Depends which virus. It can be present, but a virus cannot live in fluid. It can float in fluid. It has to be inside the cell to replicate. And then comes the debate, if it's replicating, it needs so many requirements of life, but is it truly alive? Could it use an components or not? That debate's been going on for decades. Outside the cell, it's not really considered alive, because it's just got some genetic material surrounded by protein and maybe lipid envelope, but it doesn't do anything else. Just float.
So that's why they exchange of fluid, is that it has to be in the body.
Yes, it does. Now, some viruses will survive on a surface for a fairly long time. Norovirus is one of them. Norovirus, they term it environmentally hardened. If you think of when you get a gastrointestinal disease outbreak on a cruise ship, well, that's a norovirus. And it will sit on the surface, dried up, in the sun, and it will survive a long time. You put HIV there, it's deadly now. So it just depends on the specific one now. STIs, they need to enter contact because they don't survive outside the body for long. Those are one chance. If you take bacteria such as Bacillus anthracis, or Sandra, it can produce endospores which are dormant, and they're highly resistant. They can live for thousands of years as an endospore, doing nothing, and they can turn back into a cell later. So it just depends on the organism.
All right, are there any more questions? Well, if not, then we're on to the and I'll try to make the recording available soon online, and I'll send it out to everyone. All right, I'll cut off the recording.
Thank you.
Thank you. Thanks, Gary.
Very interesting.
Yeah.